Hello students, welcome to the chemistry sessions. So I hope you all are doing good. And have you revised what all we did in the last class? So what was that? We were discussing about chemistry in everyday life, the chemicals that actually ease our work, that actually treats our diseases. The medicines that we use, those are nothing but the chemicals. So we were on to the discussion of drugs. So how do drugs actually help in a therapeutic action? That means they soothe our body, they actually treats our disease, they lower down the pain, they diminish the pain. So what kind of drugs are those? Those are nothing but the chemicals that we have studied so far belonging to the chemistry. So basically what all is included in the classification of drugs? How can we classify the different classes? So drugs is nothing but a chemical. Why we call it as a drug? Because it can be useful or it can have adverse effects as well. So basically the classification has got on the type of drugs, the chemical structure of the drug, or maybe the activity of the drug, maybe the target of the drug. So what we were on to was the target method of the drug. So drug has a prime target and it acts upon that in order to actually treat that very area, treat that condition which is actually underlying condition of a disease within our body. So that is what the action of drug is. So we were discussing about the enzymatic action. How do enzymes work within our body? So how does they work? I hope you all have revised the concept. So basically enzymes are actually having a certain kind of site within them that is known as the reactive site. So that reactive site attracts the substrate, attracts the reactant which is involved within the chemical process, within the biological process that is happening in our body, and that substrate, that reactant gets attached onto the enzyme, that active site of the enzyme, and finally we get our resultant product. So that is how the actual enzymes work. And what about whenever we introduce a drug? Whenever we introduce a drug in order to inhibit the action of the enzyme. So let us just revise that. So whenever we are having enzyme drug interaction, so what do we have in that? We have got action of drug. First of all, it is acting as a competitive inhibitor. So it is acting as competitive inhibitor as well as incompetitive inhibitor. So these terms I did not introduce in the last session. It is nothing but what we have discussed so far, the mechanisms that we have discussed so far in the case of enzyme drug interaction particularly. So the other work, the other kind of an interaction of the drug is as incompetitive inhibitor. So let us just revise the two processes that we studied in the last session. So basically what is competitive inhibitor? That means it is competing with the active site. So basically yes, we have done this before. It is, so basically the drug is competing for active site. That is what we have studied. Actually it is mimicking, it is actually mimicking the shape, mimicking the type of what kind of a substrate will that be. So instead of that substrate, it firstly attaches onto the active site and is inhibiting the substrate to get attached. It is preventing the substrate to have a reaction with the enzyme. So that is how it is going to work over here, acting as competitive. It is serving as a competition to the substrate. So I hope that is clear to you. Now in the next part is incompetitive inhibitor. Over here it is not actually mimicking, but what it does is it is going to attach on some other site of that enzyme. It is not actually touching the active site but some other kind of site. What kind of site will that be? It is allosteric site. So here it is moving on to the allosteric site, and due to this very kind of attachment, what is happening? The active site, the shape of the active site is somewhat changed. So the shape of active site is changed. So since the shape of the active site is changed, now there won't be any lock and key model. That means the substrate will not go and attach with the active site, because the substrate has not that kind of a shape, it has not got that kind of an actually the attachment which will go on to the active site. So I hope that is very well clear to you. So that is in competitive inhibitor. So it is acting as two kinds of inhibitors. First is competitive, next is the incompetitive. I hope that is very well clear to you. Now let's move on to what we have done was the receptor drug. So receptor drug interaction. So as I told you in the last class, what is the receptor doing? So receptor is, let us draw that again. So I have got here first of all receptor drug interaction that I am going to tell you. So as I told you in the last class, what is happening, how the receptor is acting. So basically there are chemical messengers within our body, they carry the messages. They carry the messages to what? To the receptors. The receptors then receive the messages and they finally transmit the message to the cell, to the prime cell. So that is how the receptor is going to work. Let me just draw the kind of interaction now it will be having with the drug. So just draw with me simultaneously. It would be something like this. First of all, I have got the receptor here. That is the kind of a receptor we are dealing with. That is the next scenario. And over here, it would be a slightly changed shape. I am going to tell you in a moment why the shape has been changed now. So just try to guess it by then. So here, this is what we have got. So this is nothing but the receptor over here. So that is the receptor. I hope you all have drawn this. Stage number 1, stage number 2 and stage number 3. So that is what we have. And what else? We need to draw the membranes. So how the receptor is attached onto the cell membrane? Its outer portion is actually lying outwards of the outer cell membrane. So here I have got the outer cells so these are nothing but the outer cell membranes, and the mouth of the receptor, actually the binding site, or I can say the active site, is lying outward of that. So here let me just draw the cell membrane first. So that is the outer cell membrane that we have, and the inner cell membrane, that is the inner cell, and the receptor is somewhat embedded within it. So these are not different kinds of receptors, but I am taking the 3 cases, 3 stages of how it is going to act. So that is how it is going to be. So this is nothing but the outer cell membrane. This is inner cell membrane. I hope you all have drawn this. Strictly I am speaking about the animal cell over here. So this is, I am speaking about the animal cell, how the messages are getting transferred over here. So this is the receptor that we have. and now there would be the action. That is the rest of the cells which are present over here. So actually that is the boundary of a particular animal cell. The boundary has got the inner cell membrane as well as the outer cell membrane. So these receptors are embedded within the membrane, having the active site in the outward portion. So here this is nothing but the active site over here, active site, or I can say the binding site. So that is how it is embedded. So let me just write it over here. The receptors are embedded in the cell membrane. Cell membrane with binding site pointing out towards the outer area, pointing outwards. So that is how it is going to be. Now what does happen? Actually now what chemical messenger will try to reach out to these receptors. So here if I draw a chemical messenger something like this. So let us just say this chemical messenger is trying to reach out over here. That is chemical messenger. It's trying to reach out for the active site. It wants to transfer the signals actually. So what will happen when it tries to attach? When it tries to attach onto the binding site, onto the active site, so finally it gets attached something like this. And that is how it is going to get attached. And during the attachment, what happens during the message transfer? This is nothing but the message transfer going on. So that is nothing but the message transfer. So during this message transfer, the shape of the receptor changes. Over here, shape of receptor changes. And what else? It is going to transfer the signals further into the cell. So this is actually, this was the outer surface, outer membrane of the animal cell, and where is the particular core of the cell? This is the animal cell over here. And finally it is going to transfer the signals which it has received from the chemical messenger onto the very core of the cell. So that is how the message is going to get transferred. It will be something like this. So that is how the message is now getting transferred. So here message is getting transferred. So that is how the process is going to be. Now what happens? Finally the chemical messenger, after transferring the message, it gets detached. So finally it gets detached over here and the receptor then regains its shape. The receptor regains the shape, and this very chemical messenger is also now detached from the site, just like in the earlier case. So that is how it is going to get detached. Got detached. So that is how the process is going to be. Let me just repeat the process, and I am also going to write the process as well. So here what will happen? First of all, I need to have an idea how the receptor is actually embedded within the cell membrane. So this is nothing but the outer walls, or I can say the cell membranes. This very membrane is the outer cell membrane of the animal cell. This is the inner cell membrane. That is how the receptor is embedded within it. And then what happens? It has got a particular binding site, a particular active site. So whenever the chemical messenger, what is the chemical messenger? It is actually a messenger, or I can say a postman, which is actually trying to transfer the messages from one point to the another. So that is actually a carrier of information. So chemical messenger is a carrier, kind of a delivery van. So it is delivering the messages from the nerves to the muscles. So here, whenever it is going to transfer the message onto the muscles, that means the cells, the very basic unit is the information is transferred to the cell, to the core. So that means first chemical messenger came and it tried to bind with the active site. So when it tried to bind the active site, the shape of the receptor changed. It took the information, it passed the information on to the core of the cell, and finally the chemical messenger got detached and the receptor regained its shape, as you can see over here. So that is the entire process of transformation, or I can say the transfer of the messages within our body. So that is how the receptors work. And now what is the drug action over here? So before going on to that, I am just writing down the key points over here for the process. So how does the process take place? How receptors work. So that is, I have got the first point over here. First key point will be, it is embedded, the receptor is embedded in the cell membrane. So that is embedded within the cell membrane, having the active site pointing outwards. So I will just write as the second point, the active site, or I can say the binding site, points outwards. What else do I need to have over here? Number 3 says now the chemical messenger comes up. So what is a chemical messenger? First I need to have the idea. Chemical messenger comes up with the information. So it came up with the information. How it came up is transferring the messages from nerves to the muscles. So message from nerves to muscles. Let me just write that over here only. So that is what chemical messenger is. First of all, it carries the information. Now the chemical messenger comes up and attaches onto the active site. So here chemical messenger binds with active site. Next would be the shape of the active site and then changes. What else? Number 6th point I am going to write over here. So then what happens? The shape of the active site changes. The message is now transferred from the receptor to the cell. So the message is transferred from receptor to cell, and finally it would be number 7th. Let me just write that properly. I hope you all have written this. Transferred from receptor, right. So the last point would be finally the message has transferred, and what we have got is the chemical messenger got detached from the receptor. The chemical messenger got detached, and what else? The last point would be the chemical messenger is now got detached and the receptor regains the active site. The receptor regains the shape of active site. So that is the entire process of how is the chemical messenger and the receptor interaction is taking place. So just note this down. Then I will be taking up the drug interaction with it. Oh, I hope you all have written this down. Now what is happening with the drug? So let us just have an idea about it. I am just rubbing this off. Now what happens whenever there is the action of drug? So again, just like in the case of enzyme drug interaction, so we have got the receptor drug interaction as well. So here what will happen? RD interaction I am writing. So what can possibly the drug do? So drug can, first of all, it can inhibit the function, block the active site. That is the first thing that it can do, acting as a competitive inhibitor. So here as well it would be acting as competitive inhibitor, as well as again incompetitive inhibitor. So here it will be incompetitive So first of all, it can do what? It can, okay, that would be over here, that is blocking the site somehow. And I may not be including that in incompetitive inhibitor. So first of all, the drug can inhibit the receptor and chemical messenger interaction. That is the first thing that it can do. That means it is blocking the binding site, it is blocking the active site over here. So it is blocking the binding site. That is the first thing it can do. And what is the second thing over here? Again, it can mimic the chemical messenger. So it can mimic the chemical messenger. So a similar kind of a method, similar kind of ways of having its prime target and actually hitting it bang on. So basically, first it would be acting as incompetitive. Incompetitive, it is just trying to block their interaction. It is trying to block the binding site somehow. So first is that included in the incompetitive, so that is for the incompetitive. And what about the competitive? Again, it is actually serving as a competition between the substrate and itself. So what is happening? What is the substrate over here? That is the chemical messenger which is going to bind on to the receptor. So basically what it's doing is, it is mimicking the functions of the chemical messenger. It's trying to act like a chemical messenger, but it is not. So that is how it is actually having an interaction with the receptor, and it may serve for a good purpose. So it may be serving a good purpose, treating the medical condition down. So this very thing over here, that is the mimicking action of, yes, mimicking action of the drug. How this can be very, very useful? So that is the question. So here I am just rubbing this off. How does mimicking action of drug is useful? So how is that useful to us? So basically, if chemical messenger, let's say for some reason it is not present in our body, the production is not going on there. So basically what we can use is instead these drugs which can mimic the action of that chemical messenger. So whenever the absence of, or I can say whenever the underproduction of the chemical messengers, we can use these drugs to mimic their action. So that will let the process go on, let the biological process will not be then hindered within our body, but it will actually go on. So what is the use? The use is whenever the, I can say the natural chemical messenger, whenever the natural chemical messenger is not produced, in the body, then the drugs having this mimicking action can be used. Then the drugs can serve as a chemical messenger. So as I told you, they are helping for the good, they are serving a purpose, they have got a purpose within our body, they are treating some kind of a condition somehow. So that is how they can treat the condition as well. So they are mimicking, it's not that they are hindering the process, definitely they are hindering, but for a good reason. What kind of a good reason? They are treating the condition, treating the actual disease which is going on in our body and relieving out our pain. So that is how the drugs are going to interact with the enzymes as well as the receptors and different kinds of biomolecules as well. They may be interacting with other kind of macromolecules present in our body in order to have a proper functioning. So that is how the drug interact. That was the basic mechanism that we have discussed. Let us just now have an idea what kind of chemical messengers are present within our body. So you must be having an idea for that as well. Just for your information I am telling these. So the type of chemical messengers, types of chemical messengers. So basically I have got two, hormones and neurotransmitters. So here I have got first hormones, second neurotransmitters. So basically, do not get confused due to the names of these. Chemical messenger is nothing but which carries the information, which carries our message within our body. That is how our body is going to respond in different conditions. If there are no chemical messengers, there won't be any kind of, actually the different kind of diseases that we have. Disease is nothing but a response of the body due to any kind of an infection, due to any kind of an ailment which is present within. So that is what appears in the form of disease. If I have got rashes, it may be due to the infection. If there are no chemical messengers, then there may not be any rashes on my skin, but the infection is still actually affecting my body and I do not get to know that. So that is how the chemical messengers work. They actually transfer the information due to which we get the indication that we have something going on in our body and we need to treat that. So that is how the chemical messengers work. First are the hormones and neurotransmitters. So that is why we call that we must be having a proper balance of hormones within our body, because actually hormones carry the proper messages. If the hormonal imbalance is there, there may be so many conditions developed within our body. So the hormones, what actually, from which gland are they secreted? So adrenal glands are there which are responsible, adrenal glands which secrete hormones. So basically I am talking about initially that was for the adrenaline basically. Endocrine glands are responsible. Endocrine glands through which the hormones get secreted first. Now what does hormone do? Hormones are nothing but actually they try to activate all the receptors within our body. So they, how do they work are, they activate the receptors within the body So those receptors which are sensitive towards these kind of hormones, then they will respond and carry the message forward. So that is how actually the mechanism is. And what else? They are not easily deactivated. That means after passing on the information, they still remains in our body. So they still remain in our body. They are not deactivated easily. And basically what is the mode of transfer? The mode of transfer is blood. They are transferred through our blood. So they are transferred, they travel through bloodstream. So that is why whenever you go to the doctor for a hormonal checkup, so it recommends a blood test for you, because the hormones are traveling through your bloodstream. So they are present in your bloodstream and there may be some kind of an ailment due to the change in the ratio of the hormones. So it must be maintained very well. In order to have a proper healthy living body, you must be having a proper balance of the hormones. What are now neurotransmitters? The nerves, the nerve endings have some kind of messages in order to pass on to these cells. So these are neurotransmitters which are actually serving as the messenger to transfer the messages from the nerves to the receptors. So the neurotransmitters, they transfer messages from nerves to the receptors. So that is how they are going to work. The neurotransmitters are going to work, as the name suggests, neuro, from the nerves, through the nerves. Transmitters, they are transmitting out the information, they are carrying out the messages. So Actually, what happens here in the neurotransmitter, I would like to serve a difference between the two. So basically, whenever the neurotransmitter carrying a message, they get attached onto the receptor binding site, what will happen? It passes on the information, then gets detached from the receptor, and finally it gets deactivated. So that is what is going on in the neurotransmitters. They are deactivated very easily. They are deactivated after passing on the information to the receptor and after getting detached from it. So I am just writing this in bracket, after getting detached from it. So that is how the neurotransmitters work. So what kind of hormones and what kind of neurotransmitters are there actually present in our body? So very most prominently what we have got here in the hormones, that is the adrenaline. What is adrenaline? You must have heard the term adrenaline rush. What is that? During the excitement, during some kind of a danger situation, you have got so much of energy, you have got so much of power that your body maybe starts shaking up, your body maybe starts sweating down. So basically what is happening, that is the release of a hormone at that situation of stress or anger or excitement or danger. So that kind of a situation releases so much of adrenaline in your body, so much of this very hormone, that all your receptors throughout the body get activated, and that is how your body gets so much of strength at that moment in order to deal with the situation. So basically that is nothing but the adrenaline rush. So some of the athletes, they try to have artificial hormones in order to have so much of power at that moment, of the sport which they are going to perform. So that is how people take advantage of these hormones as well. They take up external amounts of hormones in order to maintain their bodies, in order to have a good skin or something like that. So basically we do not want an external things going on in our body. We must maintain a healthy diet in order to have proper balance of these hormones. So basically here I am dealing with the adrenaline. So under the examples of hormones, let me just write it over here itself. Example is adrenaline. And what about the neurotransmitters? I have got two examples for you over here. One is the dopamine and the other is serotonin. And what about the structures for these three kind of chemical messengers? So for adrenaline, the structure is something like this. That is again OH and NHME. NH, yes, right. So that is nothing but the adrenaline. So that is how it looks like. It's an aromatic ring having two OH over here, and I have got OH another over here. This is represented as a dash, sorry, a wedge. So that is how we are going to represent this very group. How does dopamine look like? You need to have a proper, actually the formation of structures, proper knowledge of the structures here. So the dopamine would be something like this. So that is how the dopamine looks like. So I hope that is very well clear to you over here. Now What is actually happy hormone? Who do you call a happy hormone? It is nothing but serotonin, the neurotransmitter that we have discussed. So that serotonin is nothing but known as the happy hormone. Why this is known as the happy hormone? That you need to find out, and the answer to this I am giving you in the next session. Till then, just first revise what all concepts that we have did, and you need to have a proper learning of these structures as well. So have a good day.